Thank you very much for the invitation. I look forward to telling you about our data resource, for the Nucleome data portal. So today I'm going to give you a short introduction to the 4D nucleome. And then I'm going to talk about the sorts of data we have and how to find the data that may be of interest to you in the portal. Then show you kind of some examples of the metadata that we have and how you can visualize data on the portal and potentially query it for data of interest to you. A little bit about downloading some data files, the introduction about dkNET, reproducibility and fairness and all that. That is totally in line with what we're doing at our data portal. So I think it's very appropriate. I'm going to mention our standardized informatics pipeline and then some other ways to explore data. So for those who aren't familiar with us, we are a common fund program funded by the NIH. The 4D Nucleome is aiming to understand the principles and the three-dimensional organization of the nucleus, how that changes in time, which is the fourth dimension, how we get the 4D and 4D nucleome, and the role that this organization plays in gene expression, cellular processes, and then how this changes during development and progression into various diseases. So here's a cartoon of kind of how we think of things. So on the larger scale in the nucleus, you have chromosome territories, interchromatin compartments, domains characterized by their association with certain structure in the nucleus. So lambda-associated domains on the periphery and NADs or nucleolus-associated domains, as well as nuclear bodies where there's a lot of transcription. And then of course the larger structures like the nucleolus. And then you can drill down and start to look at individual chromosome territories. And you can largely divide chromatin into A compartments, which are regions of active transcription, and B compartments, which are your more compact heterochromatin generally. And these comprise various topologically associated domains or so-called TADs, okay? And these are regulated by a number of proteins, potentially the most important of which is CTCF. And then you can drill down even further where you get into the interesting things about gene regulation, transcription. So you can have contact between enhancers, super enhancers, things like enhancer hubs, and one or more genes. So researchers of the 4D nucleome are looking at these sort of various levels of nuclear organization in various ways. So common fund programs have the potential to be funded for 10 years. We had phase one. Our first phase was focused primarily on technological development and determining, establishing some benchmarks and things like that. The second phase is more focused on human health, disease, and development. And we've also transitioned quite a bit from bulk assays to more single cell technologies, as has much of the field. So we have two major different modalities of data that we have. We have sequencing or omics data and imaging data. And that sort of makes us somewhat unique. And we have both types of data trying to support the same sort of, answering the same sort of questions. So for sequencing data, this is kind of where things started. The workhorse is chromatin conformation capture, or more traditionally or colloquially known as Hi-C. So you can see on the right-hand side on the left, we have the various flavors of Hi-C that are done, and more variations are being developed all the time. So in situ Hi-C, dilution Hi-C, DNase Hi-C. These are kind of variations that use slightly different enzymes or techniques to get at the data. And there are also enrichment sort of Hi-Cs that are focusing on regions of the chromosomes that are contacting particular proteins or particular sequences. And what these Hi-C type experiments get you is regions of the genome that are in contact with each other, even though they're separated distantly in space or along the linear part of the chromatin. And the typical way you can view, or atypical way you can view this data, are these two-dimensional contact maps. So you have each of the chromosomes lined up both vertically and horizontally. And so on the diagonal, that's where linear parts of the DNA the chromosomes are right together. So you see this line on the diagonal. But then the interesting things are these contacts where regions on maybe not even the same chromatin or within the chromatin are contacting in regions that are far away. During the 40 nucleome first phase and continuing in the second phase, there have been several other assays developed that are in the same spirit, but they allow they're ligation-free methods that allow capture of multi-way contacts, and also different methods that allow capture of contacts between different molecules, so RNA and DNA, for example, in MARGI. As well as these chromatin conformation capture, CCC data sets, we have a bunch of other genomic data sets that provide supporting data that's also help tease apart the chromatin structure, the genome structure. So there's certain assays that can tell you the proximity to various compartments or proteins. So DMID-seq is good for laminin and centromere and nucleolar distances, same for TSA-seq. Things that look at binding of protein on the DNA, cut and run and chip-seq. And then we even have some RNA-seq data sets for expression of genes, as well as open chromatin as ATAC-seq. So we have many different types of genomics data sets. We also have a number of imaging data types. Especially during the second phase, there's been a focus on chromatin tracing, or we call it multiplex FISH experiments that essentially allow you to walk along a chromosome using hybridization, sequential rounds of hybridization of oligonucleotides to visualize the actual structure and organization of the chromatin. So we have a number of data sets of that type, and I'll say a little bit more about that later. But we also have additional imaging assays, including FISH, immunofluorescence, single particle tracking, and others. So I'm going to now go right into, oops, sorry, a demo. Okay, so we're going to try a live demo here. Here is the homepage of the 4DN data portal. Okay, right at the offering, you can see that we list the number of data sets that we have. They're comprised of replicate experiments, so we indicate the number of experiments and how many files are associated with these. And front and center on our homepage is this bar graph. And this is one way you can navigate the data, as well as provides a kind of an overview of the data that we have. By default, we group the information by organisms. So you can see we have primarily human and mouse data, but there's also data from another number of other organisms. The x-axis is experiment types. So you can see we have a large number of experiment types, and the y-axis is the number of data sets for each type. You can change this, and we could look at, for example, the number of files by lab if you're more interested in the number of files. So right here, you're able to find a lot about the amount of, or the types of data and the amount of data in the portal. And you can actually, by hovering over and clicking on the bars, you can actually go to, if there's a particular data set of interest. So if you're interested in all the chip-seq experiments for mouse, for example, click on this bar, and then you can go and browse those data sets. Also, you can do the same. So if you're interested in just fruit fly data for whatever reason, you click on that, and then you can go and browse those experiments. Okay, so that is one way to get at the data, but we try to provide several ways to get at our data sets. So you can browse all the data. You can browse by publication. And we've tried to make some collections of data that we think people would find useful. So you can go and look at all the Hi-C data sets on the portal, and these are grouped by those from a single time point and condition, time course, disrupted and atypical cells. And we also have a table of high resolution, Hi-C data sets that are those that have over 1 billion reads for the data. We also have, if you're interested in imaging, chromatin tracing data, we have a collection of those. And so we have 105 data sets of chromatin tracing data, which is perhaps, although I'm not sure, one of the, the highest amount of available data of this type. The nice thing about this data is that there are processed results available in a standardized format. And I'm not sure how familiar you are with imaging data, but it's a little bit of the Wild West as far as getting standardized formats and getting enough metadata to really understand the data. So the 4DN DCIC, especially, we have a component at UMass Medical School directed by Caterina Strabio Castillo, who has spearheaded the development of this format, standardized format. It's a multi-table format, and you can read about it here. There's a read the docs that's linked from the dataset page, as well as other places on the portal. One other thing I want to point out is that every page you go to will have this toolbar here, which allows you to browse data of various types, shows the tools, resources, and help. Okay, and I'll say a little bit more about that. But there's also the search bar. So it's another way that can help you try to find the data you're interested in. But I do want to point out that this is just a simple text search. So for example, if I put in hamster, which you may be interested in because it was used in a publication about the olfactory system and the effects of COVID. It's used as an olfactory model organism quite a bit. But so I typed in hamster, and as you see, there's the 16 hamster data sets that we have in the portal, but also 27 human data sets show up as well. And that's just because it's a simple text search. And the human data sets were also from the same paper. And the paper had, in the abstract mentions hamster. So therefore, those showed up. So when possible, we recommend using our faceted search, which I'm going to go into now. So I'll go to data browse all. Okay, and you get a more condensed version of the graph, but now you have a list of the available data sets. Okay, within this list, you can see that there are several columns of information. Each data set is a succession, the type of experiment the biosample is from. Data set and condition fields allow you to kind of group and understand a little bit more about the data, what lab it's from, and some assay details. Those are the default columns, but you're able to add additional ones if you're interested in them. So if you want to know if there have been any genomic modifications, for example, CRISPR experiments, you can go ahead and click that and that'll show up. If you want to know how many experiments are there for each data set, et cetera, these all can be chosen and configured. There's also, you can sort the columns. So you can select your different column to sort and then sort by that column. The default sort is date created. So the most recent data sets are shown first. But this is very configurable as well. You can also sort by just clicking here, and that'll give you an ascending or descending sort based on the column that is shown. Okay. So most useful though is our faceted searching. Okay, and some of these facets are, pretty straightforward. So what lab is the data produced in? Experiment type, so what's the assay? One thing I want to point out about this is, so we have like five types of bulk high-C genome wide that we have. You can select any particular one, so like in situ high-C, or you can select all high-C assays. Okay, same for fish assays, DNA fish, multiplex fish, etc. So that hopefully is useful. But there are also other things that may not be so immediately obvious, like sample type. So say if you're only interested in getting data from tissues, okay, homogenized tissues. So we have 149 data sets available for homogenized tissue. There are various, there's some high-C, there's some rna-seq, single particle tracking. And all these then get shown and displayed in the chart at the top. You can also look for your organ system of choice. So a lot of the work in the second phase of the 40N has been focused on kind of brain development and parts of the brain. So there's several brain data sets. This is only a subset of data sets, remember, because I selected tissue at the moment. We have brain and sensory system, etc. So you can drill down that way. Or if you want to get even more specific, you can go to tissue source and you can see, and you have more specific terms like forebrain, olfactory epithelium, etc. So this faceted browsing really provides a pretty powerful and convenient way to get to the data sets that may be of interest to you. So I'm just going to go start drilling down, get some data from the Decker lab, some in situ high C, and then sample sample type, sorry, stem cells. Okay, and so now I've drilled down to 12 data sets, okay? And I want to focus now to show you kind of the metadata that we have for each of our data sets and kind of how we represent it and how you can find out more information by looking at this particular in situ high C experiment, which is on a stem cell line, H1 stem cell line, human stem cell line, and it has a high level of coverage, so quite a few reads. So from this page, you can see you can get the accession. You can see if there's a publication associated. So this is the source publication. For many of our assay types, we actually have these little pop-up or drop-down windows that kind of describe the assay in case you might not be familiar with it. So you can learn a little bit about that. And then we have some basic information here, like the sample, the experiment type, and you can others information that you can click on the links and learn more about it. Then we have these tab sections of the reports down here. By default, for those data sets that have a standardized processing pipeline, you're shown the process files or the analysis result by default. So this data set has 10 process files. Several of them are associated with multiple experiments. Some of the files are associated with either of the two biological replicates associated with this data set. Scroll a little bit further down the page, you can see that these tables of quality metrics that can help you to assess the quality of the data and whether there's something you might be interested in looking at. And so for high C data sets and other types, but for those with contact matrices or these two-dimensional representations, you can visualize them right here. This is using our high-glass browser functionality that allows display of these contact maps, as well as other more traditional one-dimensional tracks or linear tracks shown above. So we have some installation score and compartments as well as gene annotations. So you can zoom around and play around right in here, and that may be sufficient. But to get a larger view and to be able to do more with it, you can go to the Explore data, okay? And from here, you can actually enter a a potential region that you're interested in, or maybe a gene. So let me just, I selected a gene and it's not, let's see. So NPHP3, which has been associated with some kidney conditions. So we can zoom in, and as you can see, once you get to the gene level, things get pretty pixely. So we'll zoom out a little bit, and you get your the resolution will kind of adjust automatically as you zoom out. Take a little bit of time, but so this region where the NPHP3 gene is located is shown here. And you can see a couple things. One, there's these typical kind of darker spots, which are regions of higher contact in this area of distant loci because they're away from the diagonal. Okay, so this might be a region of interest that you might want to explore further. And if you look, you see that there's a, if you look at the compartments, you see that this NPHP3 is kind of right at the boundary between an A and a B compartment. So we have compartment calls here. And in this data set, it seems right on a boundary. So this might be a gene that you might want to look at in the context of its genome organization and think about what that might mean. So this browsing allows you to do that. So this high gloss view is also very flexible, and you can add additional data sets. And so let's add another. So I'm again using faceting. I'm going to look for other contact matrices. So now I've got all our contact matrices. And I'll just kind of arbitrarily choose the first one and we'll apply. And now it appears right next to your other view. And you can see this data set is much sparser, but you can still see the potentially interesting things going on here. And when you zoom and pan, the two views move together, okay? You can not only add 2D tracks, but you can also add additional one-dimensional tracks. So if you want to look at some, open chromatin ATAC-seq data here, you would be able to do that. You can also save or clone these views, but you can only do that if you create an account on the portal and you log in. So I just want to, and there's a couple other things that require a login on the portal. So I wanted to just go over that quickly. So hit login, and you're currently able to sign in with both GitHub or a linked GitHub account or linked Google account, but I encourage you to use a Google account at the moment because we are going to retire the GitHub login because we're integrating authentication with the NIH RAS authorization or authentication system. So you'll be able to use ERA or login.gov to log in, as well as Google. But GitHub will go away. Sign in with Google. I'm gonna go ahead and sign in. And now it shows that I'm signing. And now I'm able to save this view if I want to, or clone this view. You can clone it to create your own new high gloss display based off this one and add it. And then you can save the links and share them with your colleagues and collaborators. So this is a very powerful system. One thing that you cannot do currently is actually upload your own files to look at them in this context. The data files that you look at have to be on the 40N data portal. However, this viewer, HighGlass, is open source, and you can set up a server on your own, and then you would be able to visualize your own data. We've had requests for data visualization in the past. We just haven't been able to prioritize it because there's some tricky bits to that. So, okay. So let's see. Let's go back here. Okay. So from this view, I should point out that you can visualize any of the files with these icons. One of these files is a Hi-C file. We provide context matrices in both Hi-C and MCOOL format. We visualize the MCOOL format in our browser, but if you visualize this file in either Juicebox or the Epigenome browser, click on these and it'll link out and bring you to that view. Okay, so there are multiple ways to visualize and navigate with these files. Okay. Before I talk about downloading files, I want to point out some other of these tabs. So you can also get the raw files if you need them. They're organized by experimental replicate. There are a number of files associated with each replicate. Another thing that you may be interested in are supplementary files, so-called additional files. And these are files that are generally provided by 40N labs to be associated with the data set. So if you remember, this particular data set was from the Thacker lab. They made their own compartment installation and loop calls and then provided those files. Okay, so they're available in the supplementary files. The Kremens lab, which is another 40N lab, did their own analysis of this data set, and they called TADs, topologically associated domains, subtats, boundaries, and dot calls. And these files are also available. So you could download any of these files for your use as well. A couple other things to point out in the attribution tab, you can see where the data sets were used, what the lab was, what the lab who generated the files were, and the award associated with it, and any publications. For now, I'm going to go back to the processed files to go over how you can actually obtain these files if you want for your own use. So there are several ways to download files. You can individually select them, okay? You can actually click on a file and then you can directly download it using this download link. However, once again, in order to download any file on the portal, you need to register at the portal and create an account to be able to download. We had to do this because we kept on getting hit by spam bots and other malicious actors trying to DDoS our portal. So having people create an account and log in, you don't have to be associated with the 40 Nucleome. You just need to register in order to download. But generally, you usually want to download more than one file. So let's say we want to download the 6 files that are associated with the data set in general. So the result of the merged and combined analysis. So those are selected. And then you just hit the button to download 6 process files. Then what happens is this pop-up window indicating that you have to have an access key in order to download the files, and it provides the command you use and kind of more information. And then you download, you hit this button to download a file of metadata about these particular files that you can then use this command. So here's what this file will look like. Let's see if we can make it a little bigger. So this is the download URL, and you just essentially get that field and use the curl command to download the files. And so you can download them in bulk. The command is again given at the top of the file here. And so it's a way to bulk download the data. Okay. You can also download directly from the browse view. So just to show you. So when you browse the data, I'm logged in now, so some of this data hasn't actually been released. So I probably shouldn't. So for the release data, you can look at the raw files, okay, shown here. And this, you can just select them to download, and their process files associated, and you can select those to download. And from the top of the page, you can select them all, and you could potentially download all the files that you wanted. So that's another way that you can download the file. Okay, so you have the data. Why why might you want this data, or what's kind of special about this data, or how is these analyses results generated? Well, you can always look at a provenance graph, and this shows the actual analysis steps of the analysis to generate the final outputs. So you start with your FASTQ, you align them, you do some post-alignment processing, you generate the matrices, and you finally call compartments and insulation boundaries, insulation scores and boundaries, and you end up with these files. Okay, so the nice thing is that all the IC data sets, and also for several other types of data on the portal, they're standardly processed, run through the same pipeline. So if you look in our resources section, we have information about data analysis, some general information about reproducible data analysis, reference files, etc. And then specific information on different pipelines. So we have the high-C processing pipeline, we have an overview, and then the individual steps, and where you can find the code. All this is open source. We have the 4DN DCIC GitHub repository, and this is the particular repo for the 4DN high-C pipeline. And we know that several people have used this to do their own analysis. So if that's your bag, you're more than welcome to do so. All right, let's see. And so not only Hi-C, but we've taken the rna-seq, attack-seq, chip-seq pipelines from the encoded project and adapted them and run so essentially the data should be analogous and comparable between encode data sets for those types of analysis pipelines. We don't have any single cell pipelines implemented, but generally we take the analysis results from the labs in those cases. Okay, the last thing I wanted to touch on as part of the demo is that we do have tools, and one of them might be useful, is the 40N Jupyter Hub. This allows you to do some exploratory analysis of the data without even downloading it. So you're essentially you can run Jupyter notebooks in the cloud. Again, you need an account for this, and it's even a little more restrictive because we've had people come and try to mine Bitcoin on our Jupyter Hub. So we don't like that. So you have to have a lab associated with your account. Now, if you're not part of the 40N, that's okay, but you need to contact us via email, say why you want to run your analysis on the Jupyter Hub, and then we can associate an external lab with you that will allow you to log in. Okay, so this essentially is a standard Jupyter Lab interface. And if we look at a particular notebook, there's some examples in the example directory. And this is an example of how you can get started using Jupyter Hub. So you can just go through that, but it's not terribly exciting, but I've done a query for 6 experiment sites and got them, and so on and so forth. And there's a few examples. One that uses previous data from a previously published paper that's on the portal to recapitulate their analysis. All right, so I then want to go back to a couple slides just to wrap up. So here's a slide of useful links. So our data portal, the data collection, I mentioned the fish omics format. All these are linked from the data portal as well. We have a high-glass tutorial video on YouTube. And there's other useful resources. I do want to point out the 40N Organizational Hub. We are the Data Coordination Center. They are the people who, they have a website that lets you know about upcoming events, has links to various standards and protocols that we use. So it may be useful to visit them as well. To find out more, we have a somewhat recent, a couple of years ago now, but data portal paper in Nature Communication. You can find it here. And finally, here's the team that made it happen. This all happens in Dr. Peter Park's lab. There I am. The top row here are the people you're most likely to interact with if you try to get in touch with us. Rahi and I are wranglers, so we help manage getting the data into the portal and providing help. And Clara's a bioinformaticist who kind of helps out with bioinformatics question. This is the infrastructure team. So there are several projects in the lab that use the same underlying infrastructure, which was taken from the encoded portal, ENCODE project. We forked their infrastructure and now use that for three of our own projects. This is our UI team who were responsible for this, making the portal look great. The high glass and some other of the visualization tools was spearheaded by Niels Skellenborg, who's another member of our department. And a lot of the imaging work was done with our UMass medical school colleagues, primarily Katerina Sombio and her colleagues. So I'm pretty sure that that's it. I'm happy to take any questions, and thank you very much.
Yeah, thank you for Andy for the great presentation, introduce us this useful resource. So now is our Q&A session. Please feel free to unmute yourself or type the question. I will ask the speaker. So there is a question from Dr. Marcus. Great project. Since the Common Fund is limited to 10 years of funding, what provisions are being made to maintain curation and access to these important data once the funding ends?
That is a great question, and one that we are, I don't want to say asking ourselves because we've thought about it as we're nearing our conclusion, but so we have several ideas and several things that we're doing. We're putting our genomics data sets into Geo. Many of them are already there. For those that aren't, we're working on exporting them to Geo, so they'll be available on Geo. For the imaging data sets, we're looking at some more general repositories like Zenodo and such. However, once you do that, you lose the queryability and a lot of the metadata organization. So what we're hoping to do is, at least for some time, have essentially, once the project ends and we have all the data in, it'll all be publicly available. We'll maintain the portal at a minimal, essentially read-only state. We are working with the Common Fund data ecosystem on other strategies and ways that we may be able to do this. We are part of the Amazon open data service. So that means that we don't have to pay for storage for our public data nor egress fees, which is a huge advantage. So all these things will help to keep the data available and hopefully queryable and usable into the future. So the CFDE, some deposition of data sets, something called the DRS service, which I'm not sure if you're familiar with, and I won't go into too much, but it's just a way to point to data no matter where it lives and allow it to be accessible. So we do have plans, and we're working hard to get those in place.
Thank you. Hope that answered the question from Dr. Marcus. If you have more, just type or unmute. The next question is from Dr. Swapandas. It will be interesting if we can query, browse, and get information on a genomic location of our interest and its interaction with other gene regulatory regions. For example, if it is a part of promoter-enhancer interaction in a particular cell type, And if that interaction get altered by some intervention, is it possible in the 4DN browser now?
Yeah, that's a great question, and it's something we've been working on. So short answer, doing exactly what you're asking is probably not that easy at the moment. However, if you remember the high glass views that I showed, you can get multiple data sets up at the same time and look at them side by side. You can query them independently for different regions. They don't have to stay locked together, so you can look at different areas. Um, and you can use a faceting searchability to look for your particular cell type or region, although the direct query of regions is not currently, I mean, it's supported, but not that straightforward at the moment. You can definitely do it in Jupyter Hub. So yeah, not a completely satisfying answer to your question, but it's something that we work towards. We haven't quite got there to make it as easy as we'd like right yet, but we can also help to do that. Oh, I do realize I forgot to show my very last slide, which I think I might have lost it. Anyways, it was to our help mail. So at the portal in the help section, there's the way to get hold of us. And the email is support@4dnucleome.org. So anytime you have a question, feel free to contact us, okay? Yeah, and I didn't mention it all because it's a bit early days, but there are efforts at both the CFDE, the Common Fund Data Ecosystem, as well as within our group in the 4D Nucleome to make associations with variants and regions and allow that to be queried. So I would say stay tuned for that. It's something that we hope is coming soon.
Yeah, and I think there's a comment from Dr. Swapandas that people working on human GWAS hits will be interested in that functionality. Thank you.
Yeah.
Okay, so hope that answered Dr. Das' question. And there is one more question from the chat. So is the data portal open to any research to submit their data, or do I have to publish paper before submit my data?
Okay, there are two questions there. So the question about can anybody submit their data? We deal with that on a case-by-case basis. We are resource limited in that we only have the two of us working on submissions where we're primarily, working with people in the consortium. They're generating a lot of data, so we aren't always able to accept external submissions. However, we have and we do, so definitely get in touch if you have data that you might want to submit. And we can talk about the process and how that could happen. And then the other question, I've lost it.
I think it's the publication is the
Oh yeah, do you need a publication? No, you don't necessarily. We actually have a cardiac data set that was submitted by a researcher who used to be part of 4D Nucleome in phase 1, and he generated the data and they didn't end up publishing it, and he submitted it and it's available. So, and that's just one example. There's several data sets that aren't yet associated with the publication, especially some of the benchmark data sets that were used for kind of benchmarking the techniques, and they're all available. So yeah, if you have data that you might want to submit, get in touch and we can determine what might be possible.